In this video, we will be talking about what is mitosis and the stages of mitosis, followed by what are the stimulators and inhibitors of mitosis. Okay, so we will begin with the topic. What is mitosis? Mitosis is a process of cell division in which one cell gives rise to two genetically identical daughter cells. So when one cell becomes two cells, we call the process of cell division as mitosis. We have two types of cell division that happens in the body. One is mitosis, the other is meiosis. Mitosis is the result of the division that happens in the somatic cells. So here the number of chromosomes remain the same in the parental as well as the daughter cells. So here we'll start with the definition of mitosis. Mitosis is the process of cell division in which one cell gives rise to two genetically identical daughter cells. So the most important point here is two genetically identical daughter cells, whereas in the other type of division, which is meiosis, we have genetically non-identical daughter cells. The number of chromosomes will become half. So this was discovered by Strasbourg. Later, W. Fleming discovered this in animal cells. The term mitosis was coined by Fleming. The number of chromosomes is preserved in both the daughter cells. So if the parental cell has 46 chromosomes, both the daughter cells have 46 chromosomes as well. So this happens in somatic cells, whereas meiosis happens in the reproductive cells. Mitosis is similar in both animals as well as in plants. Why does our body take mitosis as a process? Mitosis is a method of cell division which is going to help in cell reproduction. It increases the number of cells, which is going to help the organism to grow. Now, whatever damage that happens to the body is going to be continuously replaced with the help of mitosis. So our cells continuously die and get replaced by the process of mitosis. So here we have regeneration also that happens with the help of mitosis, and mitosis acts as a method of asexual reproduction in most of the organisms. So starting with the stages of mitosis, so cell division happens starting with interphase. Then we have the mitotic stages. We have prophase, late prophase, metaphase, anaphase, telophase, and cytokinesis. So we will start with the first stage. The stage that happens before the mitosis is the interphase. Interphase, we have the S phase of interphase. In interphase, we have three stages. We have the S phase, the G1 phase, the S phase, and the G2 phase. So here we have the process of in S phase that happens before mitosis. What happens in S phase? In S phase, we have the process of DNA replication that happens. So interphase is a part of the cell cycle. So here we have the copies of the DNA that happen before the M phase. So the DNA has to be made into a copy before they get separated during the process of mitosis. So here this interphase has three phases: G1, S, and G2. So this interphase is a stage where the cell prepares for cell division. So here we have enlargement of the cell, we have the duplication of the DNA, and other process that happen for the formation of cell division. The first stage, the first stage in mitosis is prophase. So prophase happens by the appearance of thin thread-like condensing chromosomes. So we have the appearance of the thread-like chromosomes that happen. So what happens next is the cell becomes in spheroid shape. The chromosomes in the prophase are composed of two coil filaments, which are the chromatids. So here what happens is after this DNA has been replicated in the prophase, they will start condensing in order to form chromosomes. The chromosomes will now have the chromatids. So as you can see, we have the early prophase, which has the formation of the chromatids. Prophase, we have the formation of the chromatids, as you can see in this image as well. We have the formation of the chromosomes in the form of chromatids. So for this to happen, the DNA has to condense. So they will become shorter and thicker. And the two chromatids are held together with the help of the centromere. The chromosomes will approach the nuclear envelope. There are centrioles that appear during the prophase. The centrioles are now going to migrate to the opposite poles of the cell. So as you can see, we have the centrioles which move to the opposite poles of the cell. The nucleolus will gradually disintegrate. Now, what happens here is the plants and animals, the nuclear envelope does not completely dissolve during mitosis. Now, the next stage is the pro-metaphase, which is the stage between prophase and metaphase. So, during this stage, we have the breakdown of the nuclear envelope. So, what we had here earlier is the chromatids present inside the nucleus. The nucleolus has disappeared. The centrioles are in the opposite sides of the cell. But when we move to the pro-metaphase, the nuclear envelope has disintegrated. And we have the interaction of the spindle fibers with the chromosomes. So the spindle fibers that are arising from the centrioles are going to attach to the chromosomes, and the region where they attach to is known as the kinetochore. The kinetochore is a region on the centromere to which the spindle fibers from centrioles are going to be attached. Now, the sister chromatids are attached to the spindle fibers. This is what happens in the prometaphase. In the metaphase, all the chromosomes are going to be arranged in the metaphase plate. The chromosomes are going to be the shortest and thickest during this phase. The chromatids are going to occupy the plane of the equator, which is known as the metaphase plate. So they are still attached to the spindle fibers through the kinetochore. You can see here the kinetochore microtubule, which is present. The kinetochore microtubule is going to be attaching to the centromere. Centromere is the yellow region in the image, which is holding the chromatids. So each chromosome has now duplicated. So we have two pairs of each. In the metaphase, the two chromatids of the chromosomes are going to repulse each other. So as we mentioned, the kinetochore is the attachment site for the spindle microtubules. So they are all arranged on the medial plate or the metaphase plate during the metaphase stage. Next is the anaphase stage. In the anaphase stage, we are going to have the synchronous splitting of the chromosome. The chromosomes which are held together by the centromere are now going to be split, and then they start moving towards the poles. So the splitting of the centromere happens due to the increase in the calcium ions. So the daughter chromosomes are the ones that are formed after the splitting, and they're going to move towards the opposite poles. So the movement of the chromatids towards the pole can be due to the shortening of the microtubules. So we have the microtubules that are coming from the spindle fibers that have attached to the centromeres, pulling them back to the poles. So the microtubules will shorten. Microtubules which are made up of tubulin dimers, they are going to be shortened in order to pull the chromatids or the chromosomes to the opposite poles. In the anaphase, we have the poleward migration where the chromosomes are going to be moving towards the poles. So as you can observe in this image, we have the centrioles which are attached to the centromeres through the microtubules. So the microtubules are going to move the chromatids to the poles. We have around 30 ATPs required to carry the chromosome to the poles. The next stage is the telophase. The telophase is when the end of the migration occurs. Now the daughter chromosomes have reached their poles, and this is the beginning of telophase. So in telophase, whatever happened in prophase, the opposite of that is going to happen in telophase. So what happens in telophase? The nuclear envelope will reassemble, the chromosomes will form the daughter nuclei. Now, after this, the mitotic apparatus is going to disappear, which is the centrioles, whereas the spindle fibers that were formed, all of that is going to disappear, and they are going to reduce the viscosity of the cytoplasm, and then RNA synthesis will take place. The chromosomes will then decondense to form their long, slender, extended form, and the nucleolus will reappear. So to revise what has happened so far, in prophase, we had the condensation of the chromosomes, we had the disappearance of the nucleolus, we had the formation of the centrioles, In the pro-metaphase, the nuclear membrane disappeared. In the metaphase, the chromosomes were all attached to the spindle fibers and they were arranged in the metaphase plate. In the anaphase, they were pulled apart to the opposite poles. And in the telophase, we have the reoccurrence of the nuclear membrane, reoccurrence of the nucleolus, and then we have all the apparatus that was used being disintegrated. So the spindle fibers are going to be disintegrated and the decondensation of the chromosomes will occur in the telophase. Now, cytokinesis happens is nothing but the division of the cytoplasm. So, division of cytoplasm is defined as a cytokinesis. It officially begins during the anaphase and it is continued through the interphase, and then it moves also after your telophase stage. So cytokinesis is the last stage of mitosis. So what happens in cytokinesis is the constriction and furrow formation in case of animals. So in animals, we have a constriction or a furrow formation that happens. We have the formation of the constriction of the plasma membrane. Here we have at right angles, the plane of at the metaphase plate, wherever we had aligned the chromosomes in the beginning, at that metaphase plate, we have the formation of the cleavage furrow. The cleavage furrow will then start to widen and then they will go to the center of the cytoplasm and then divide the cell into two cells. So here we have the metaphase plate at right angles. We have a constriction which is going to grow deeper and deeper, which gives rise to two daughter cells. So this is what happens in the animal cells. Whereas in the plant cells, we have the formation of a cell plate. So what is the cell plate? So the cell plate is a formation of a layer in between the karyokinetic nuclei. So what is karyokinesis? Karyokinesis is a division of the nucleus. So once the nucleus has divided into into the two daughter nuclei, we have the formation of the cell plate. So cell plate is a formation of a specialized structure called as a phragmoplast. So phragmoplast consists of lots of microtubules and actin filaments that is appearing in the center of the cell. So after the formation of the two daughter nuclei, we have the formation of phragmoplast. So phragmoplast is a specialized cell organelle that is forming. It consists of lots of microtubules and actin filaments, and it is appearing at the metaphase plate between the two daughter nuclei. So this is going to guide the vesicles that come from the Golgi. So what we have in the cell plate, phragmoplast. So after the phragmoplast is formed, we have vesicles that come from the Golgi. The Golgi will synthesize the lipids and the cell wall polysaccharides and other components that are required for the cell wall formation and send it to the phragmoplast. The phragmoplast will now become bigger and bigger. It will accumulate all the vesicles coming from the Golgi, which are also known as the dictyosomes, and then it will start forming the cell wall at the metaphase plate. So this formation of the vesicles along with the phragmoplast forms a disc-shaped structure which is known as a cell plate. The phragmoplast will now expand as and when the new Golgi are getting added and the cell plate is being formed. The growing cell plate will now grow towards the metaphase plate and separate the newly formed nuclei. So now we also have the formation of the plasmodesmata that happens during the cytokinesis stage. So the smooth endoplasmic reticulum, some segments of it are trapped within the growing cell plate, which forms the plasmodesmata. So now we have the parental plasma membrane and the parental cell wall, which is growing deeper and deeper, giving rise to the formation of two new cells. So formation of the plasmodesmata is a channel between two cytoplasms. This is happening because of the segments of the smooth endoplasmic reticulum that get trapped within the cell plate. So now we have the cell plate that is formed, and now the cell plate is now passing through the cytoplasm. It now gets transformed into the primary cell wall for the two new daughter cells. So as you can see, these are the two new daughter cells. In case of the animal cell, we just had the cleavage furrow which deepens, whereas in the plant cell, we have the formation of the cell plate, which is happening because of the presence of phragmoplast. Phragmoplast consists of lots of microtubules and actin filaments, and we have the Golgi apparatus sending vesicles from everywhere and forming the cell plate. Now, the significance of mitosis in the cell, it helps in genetic stability. It helps in the overall growth and development of the organism because mitosis helps in increasing the number of cells. It helps in tissue repair and regeneration. So whatever old cells are there in the body are going to be replaced and regenerated by the process of cell division. It is a method of asexual reproduction in single cell organisms such as the yeast. It also helps in maintenance of the chromosome number. As I said, if the parental cell has 46 chromosomes, the daughter cell also has 46 chromosomes. We can use mitosis for producing clones. We can use mitosis for the process of tissue culture. We can use mitosis for stem cell regeneration. Now, coming to the mitotic stimulators, the processes in the cell that are going to activate mitosis, we are going to have the first one as growth factors. So, growth factors are proteins that bind to specific receptors in the cell membrane, and they're going to activate the process of cell division. They're going to help the cell progress in cell cycle. So, cell cycle we have the G1, we have the S, we have the G2, then we have the M. So, for the cell to move to the cell cycle, we have the proteins which are known as the growth factors. So, growth factors will activate something called as the tyrosine kinase receptors. So, we have examples of growth factors. So, we have the different signaling pathways which are not necessary right now. We will just have to look into what are the processes that activate mitosis, mitotic stimulators. The first one is growth factors. We have epidermal growth factors and platelet derived growth factors. So epidermal growth factors is going to stimulate DNA synthesis and cell proliferation. Platelet derived growth factor is going to promote wound healing and activate the cell division. So the process of cell division is being activated by the presence of these proteins called as growth factors. We also have the hepatocyte growth factor, which acts on the hepatocytes, which promotes the liver regeneration. We have insulin-like growth factor, which is going to act on multiple types of cells, which is going to promote cell proliferation. The fibroblast growth factor is going to act on fibroblast. It stimulates angiogenesis, which is the formation of new blood vessels and tissue repair. Next is a vascular endothelial growth factor, which works on endothelial cells and it promotes the mitosis during new blood cell formation. So these are the different growth factors. Growth factors are the proteins that are going to activate the process of cell division. The next type of mitotic stimulators are hormones. Hormones are going to promote mitosis by targeting the tissues. We have estrogen. Estrogen works on uterine and mammary glands and they promote the proliferation of the cell during the menstrual cycle and puberty. The thyroid hormones they are going to work on many tissues. They are responsible for the increase in metabolic rate and the overall growth of the organism. We next have the growth hormone. Growth hormone works on liver and other tissues and they stimulate the process of mitosis by activating the IGF. IGF is insulin-like growth factor. Erythropoietin works on the bone marrow and stimulates the erythrocyte precursor cell division. Next, we have cytokines. Cytokines are going to work on the immune cells and activate the proliferation of immune cells. So the activation of mitosis can also come from hormones. The third is substances known as mitogens. Mitogens are substances which are usually proteins. They're going to trigger the cell division by pushing the cell to move from the G1 to the S phase. So mitogens will bind to the cell surface receptors. We have the signaling pathway, which is known as a RAS-MAPK signaling pathway. This is going to induce cyclin D expression. Cyclin D is going to activate CDK4 and 6. CDK4 and 6 is going to phosphorylate RB. RB is going to release a transcription factor, which is going to activate DNA replication. So this is an example of a cell signaling pathway that is activated by a mitogen, and the end result is nothing but the replication of DNA. So we start with a mitogen. Mitogen binds to its receptor. This is going to activate the RAS-MAPK pathway, which is going to induce cyclin D expression. Cyclin D is a cell cycle regulator protein. The cyclin D activation is going to lead to the activation of CDK4 and 6. CDK4 and 6 is going to activate the RB protein. The RB protein is going to release the transcription factor E2F. The transcription factor is going to activate DNA replication. So examples of mitogens, we have 5-timer-gluten, we have lipopolysaccharides and lectins. So these are all examples of mitogens that are going to activate the process of mitosis. We also have nutrition and environmental stimuli, which are nothing but the external factors that are going to trigger cell division, such as adequate nutrients. If more nutrients are there, cells are more likely to divide. We also have oxygen, temperature, and pH at optimum levels can act as stimulators for mitosis. Now, moving to inhibitors of mitosis, we have the first process which is going to block mitosis as physiological inhibitors. So here we have contact inhibition, differentiation signals, tumor suppressor genes, and cellular senescence factors. So when contact inhibition is nothing but the density of the cell. So in a particular region of our body, when the crowding of the cells start, the cell division is going to stop. So the process of more number of cells being present in a region is going to send a signal that the density has reached its limit, and when density has reached its limit, contact inhibition occurs and mitosis is being stopped. Next, we have differentiation signals. Whenever a cell is getting differentiated, which means, so when all of us we have all started from a single cell, and this cell has divided into multiple cells, and each cell has carried out its own function. For example, some cells have become nerve cells, some cells have become muscle cells, some have become our organelle cells, some have become our blood cells. So once they have acquired their specific function and structure, we call that process as differentiation. When differentiation has occurred, the particular cell will stop dividing. So cell division or the process of cell cycle is going to be halted when the differentiation is going to start. So differentiated cells will exit the cell cycle. Tumor suppressor genes. So these are genes that are going to stop the process of tumor formation, and therefore they are going to stop the process of cell cycle. So cell cycle inhibitors, we have the examples under physiological mitotic inhibitors. The last is cell senescence factors. So whenever a cell has aged, whenever a cell has reached its limit, we are going to push the cell towards senescence and further into cell death, and therefore the cell will stop dividing. So whenever a cell reaches the Hayflick limit is the number of cell divisions that the cell is allowed to have after which it is considered as an aged cell. Next is chemical and drug-based mitotic inhibitors. We can stop the process of cell division using certain chemicals and drugs. We can basically stop the process of mitotic spindle formation. We can stop the DNA replication, and we can stop the enzymes required for this process in order to inhibit mitosis. So the first is microtubule targeting drugs. These are drugs that are going to stop the process of the microtubule formation, that is helping the cell division process. So we saw microtubules attaching to the centromeres and pulling the chromosomes apart. So that microtubule formation is going to be halted for us to stop the process of mitosis. So first is coelokine, which it is a drug which is taken from the autumn crocus. So this is going to bind to the tubulin and prevent the microtubule polymerization, which is going to block the metaphase. Next, we have aurora kinase inhibitors. So these are going to be stopping the mitotic spindle formation and chromosome segregation. So we can form drugs and chemicals that are going to stop the microtubules from forming. And if no microtubules are there, the chromosomes cannot be separated, and therefore cell division does not occur. Next, we can stop DNA synthesis and topoisomerase inhibitors. We are going to stop the process of DNA replication by using drugs such as doxorubicin. Doxorubicin is a DNA intercalator, so it's going to bind in between the DNA strands and then not allow it to replicate. Next, we have alkylating agents. So alkylating agents, these are going to form covalent bonds with the DNA, and they're going to prevent the replication, and therefore they're also going to prevent transcription. So we can stop mitosis by using drugs that bind to the DNA molecule and prevent it from getting replicated. If replication has not occurred, we do not have the DNA to get separated during cell division. So cell division is halted. The third type of inhibitor, we have physical inhibitors. So radiation will cause DNA damage, leading to cell cycle arrest. Temperature extremes will denature the proteins that are required for the process of cell division. Example, the mitotic spindles. Hypoxia, which is low oxygen, can also inhibit ATP production and halt the mitotic energy dependent processes. So we have the inhibitors that started from the physiological inhibitors, where the natural process of growth in the organism is going to halt cell division. Next, we had the chemicals that we can use in order to stop cell division. And we have the physical reasons that can stop the process of cell division. So with this, we have completed the topic of mitosis, the stages of mitosis, what are the processes that are stimulating mitosis, and what are the processes that are inhibiting mitosis. So thank you so much for listening.